you've also seen this, and you know that we only find a variant in about 60% of patients that we screen those 7 genes for. And that's great, but there's also 44% of patients where we don't find anything. And this is where we're going to kind of get to what I'm going to talk about. And so we know it's complex, and we also think there are other genes that are involved. And so what we did was we created a new platform for novel gene discovery in our lab. And typically what Carla does in the lab when you send your blood in and she screens each gene individually. And this can take a lot of time, and that's why it takes about 3 months for you to get your results back. And a lot of the time people will only request factor H because it's the most commonly mutated and never get screened for the other 7 or 6 genes. And we know that you can have variants in more than one gene. And so we kind of wanted, the idea was to, let's do it all at once. Let's save money, save time, and give you as much information as we possibly can. And so we designed this platform, and there's actually been 2 versions, and I included information on both versions because I'm going to show you data from both versions. And so the first version, we had 68 genes that we were going to screen in one experiment, and that's about 300,000 base pairs. And so just to give you a reference, when Carla does one reaction for one part of a gene, now she can only cover about 300 to 500 base pairs each time. And so we're doing over 300,000 in one experiment. So it's a great deal more. And then in our second version, we actually found we could increase the amount of genes that we wanted to screen and make it more comprehensive. And so we have all been talking a lot about the complement cascade, and we know the alternative pathways involved in this disease, but in your body, nothing works alone, right? And everything is kind of working in parallel to make you the healthiest individual that it can do. And so we know that the coagulation pathway, which we've kind of already discussed with ADAM-TS13 is a part of the coagulation pathway, is very intimately involved with the with complement. And so we included genes in the coagulation pathway. So the first pie graph is version 1, and you can see that we had the toll-like receptors, but it's not important for you to ever learn those because we took those off the platform because we didn't find anything, and they were really hard to capture using this kind of technology. And so instead what we did in version 2, which is the second pie chart, is we just upped the number of genes in the coagulation pathway. So now we have about 68% of the genes on the platform are part of the complement, and that includes the classical, the lectin, and the alternative pathway, as well as the terminal complement that actually forms the MAC and lyses a cell. And we also included several genes in the coagulation pathway. And so here's just a really quick view of all of the genes that are included. So here's the alternative pathway. Here's the terminal. All the genes in red, I don't know if that's hard to see, are genes that we know are involved. Here are all the regulators of the alternative pathway. Whoops.